Hello, welcome to Science Hangout from Science Mission. This is our first interview. We have today Professor Nikolay Dokholyan from University of North Carolina at Chapel Hill. Welcome, Professor.
Hello, thank you for having me.
Many of you might be familiar with the professor, but some of you may not be aware of his accomplishment. I'm going to briefly tell you the biography of the professor. Dr. Dokholyan holds Michael Hooker Distinguished Professorship since 2014. He is an American Physical Society Fellow since 2013. Professor Nikolay Dokholyan was awarded March of Dimes Basil O'Connor Starter Scholar Research Award from 2004 to 2006. He also received the University of North Carolina Chapel Hill IBM Junior Faculty Development Award in 2004. He is a recipient of UNC Research Council Award in 2003. Dr. Dokholyan received NIH Postdoctoral Fellowship from 1999 to 2002. Thanks for joining us at our Science Hangout, Dr. Dokholyan. Let's get to some of the questions about yourself. Can you please tell us about yourself, your background, education, research projects, etc.?
Sure. So I grew up in a former Soviet republic called Georgia. I got my interested in science there. I went to physics mathematics school, and that determined my path to be. I wanted to be a physicist based on that, and I went to Moscow Institute of Physics and Technology. Came to United States and working on in high energy field, but because there was a huge outflux from that field, super collider was closed. A lot of scientists changed their directions, and I wanted to stay in science, and I really liked biological physics. So I started studying protein folding, and I started with protein folding. At that time, it was a very exciting field, and by beginning of 2000, many things about our knowledge about protein folding has been, to some extent, teased out to many regards, and I got interested in misfolding of proteins and something that results in disease, so the processes that result in the disease. So when I started my lab at UNC, I basically focused on a disease called amyotropic lateral sclerosis, but also other misfolding diseases such as cystic fibrosis and Alzheimer's and Huntington and so on. So a lot of neurodegenerative diseases have this common property where proteins misfold and tend to aggregate. And whether this aggregation has anything to do with the disease is still unclear, but the aggregation processes are definitely associated with the disease.
Okay, so how was this transition from physics to biology? Was it difficult, or like it was a smooth transition? How much of
yeah, it was painful, but it was a good one. I mean, it's like starting your life again. It was very interesting. I remember I basically started taking undergraduate classes in biology. That's how it all started. And then I basically read books on my own. And it was a lot of work, that's for sure, because I came from the field of high energy physics. It's so theoretical that I didn't really know what proteins were. It just was really some abstract concept to me. But the world of biology is so fascinating. It's just so different and so interesting. And there were so many questions that you could not even pose. Like in physics, you often have a question and then you try and struggle to address it. But in biology, posing a question is probably 90% of the deal. I mean, it was hard. And so it was very interesting. But the learning was all the way through my life, actually, because I started learning biology and biophysics as a graduate student at the end of my graduate school. And then when I came to UNC, I quickly learned that I want to actually test my predictions, and I always was computational or theoretical person.
Uh-huh.
Testing predictions means you want to have either experimental collaborator who would do it for you,
Yep.
or which is less likely if you're an assistant professor. So I decided to learn experiments myself. So I spent a lot of nights in the lab learning pipette, and it was quite an interesting experience too.
Wow. Wow. Yeah, I know. Like biology, like one experiment works, next experiment doesn't work. It's kind of it's not straightforward thing in biology.
Nothing is straightforward, and that's the beauty of it.
Yeah. Yeah.
It depends how you take it. Yeah.
Yeah. You might have spent very long hours in the lab learning and all those things.
Right. Yeah. I mean, I was lucky with incredibly good students.
Uh-huh.
So my first graduate student was Sagar, who basically was learning experimental biology alongside. I mean, actually, he was doing most of it, and I was just more like shadowing him.
Uh-huh.
Amazing collaborators in the department. Department is very unique, I would say, in terms of how people are connected with each other, how they're collaborative. So I started working with a former colleague of mine, Michael Kaplov, who was teaching us a lot of techniques, who gave us part of his lab to do a lot of experiments before he retired. So that's how we started.
Okay. That's interesting.
Yeah,
And
2003.
2003. Yeah. Yeah, it's about 10, 12 years ago. So have you thought about when you are in the academics during this transition to go to the industry or stick to the academics? What made you stick to the academics?
Freedom. I really like I really like working on questions that I like to ask. And so in industry, it would not likely not be the case. I have had offers from industry, but it's hard for me to give up the freedom, and so I decided to stay in academia. And I have to say, I mean, when you say freedom, it doesn't mean that you are completely free. You have to really think about the projects that you do. You may, and you have to convince the funding agencies that what you do is actually really cool and beneficial to the society. And you have to convince yourself that it's worthy, of course. But if you can do that, then you have complete freedom how you approach things, and that's really amazing.
Yeah, that is like you see most of the breakthroughs coming from the academics rather than from the industry.
Industry also has a lot of resources, and they have very smart people too. So
No, I mean, like, I'm not saying that they're not bad or anything like that, but I mean, like, most of the research, like basic research, science research, comes from the academics rather than the industry. So, like, do you have any role model or anything like whom you look up to, a mentor or somebody in your career so far?
I had a lot of good role models in science, and I have to mention some of them are really remarkable people, and there are quite a few of them actually. And one of them I would like to particularly mention. His name is Michael Levitt, who is Nobel laureate in computational. He developed molecular dynamics, one of the developers of molecular dynamics. But he is at the same time, and besides the fact that he's an incredible scientist, but he is such an incredible person as well, very humble and very wise, and it's very interesting to interact with him every time I get a chance. So it's really great. And but not only Michael, there are many other people who I really admire for different aspects of their research and personalities. But what I value in science is basically purity. It's the science is all supposed to be pure, right? Apolitical, all about finding the truths of figuring out how things work, and you can't lie or can't make partial truths about it, right or wrong. You just don't know the rest.
Yep, that's good. And like you have, do you have to take classes like for teach undergrads and all those things? So how do you like it, if at all?
So whom do I teach?
Yeah,
Or
like whom do you teach, and how is it going? Do you like that aspect of the
yeah, I like
academic?
teaching since even an undergrad. So I was at I was even back when I was an undergrad in Moscow. I was one of the teachers for uh, the physics mathematics school, evening physics mathematics school, where kids go after school and do extra math and physics, and we would teach them cool
Uh-huh.
things to do, and
Yeah.
it was really fun. So I really like teaching. What I teach now is one of the modules in biophysics. So we have
Mhm.
really an outstanding program at UNC in biophysics. It's one of the oldest in the country, and it has a remarkable curriculum. And
Mhm.
actually, it's the reason why I joined UNC when I was looking for a job. I had some offers, and I decided to go to UNC because of the graduate program in biophysics that they
Uh-huh.
had here. And that it's more like a boot camp for students.
Uh-huh.
It's
Yeah.
an elite program, so you cannot get really PhD in biophysical program. You can only be part of it, and you get
Uh-huh.
like on your PhD statement that you have specification in biophysics, and you completed this program. But it's really an elite program, and it's hard to survive. But if you survive there, it's really tells something about you.
Wow.
Yeah, so I'm teaching one of the modules in this course,
Uh-huh.
occupied with protein folding, misfolding, and kinetics of chemical reactions, stochastic processes that happen in
Wow.
cells. So it's an intense course.
Okay. And how do you spend like I'm not sure whether you get any spare time to do other things, and like would like to know your hobbies and what are your interests and all those things. Like do you,
Well, I have 4 kids, so with that you get to do a lot of activities,
uh-huh.
drive them, meaning driving them around. But
Yeah.
I find time for myself. I like cycling, so that's one big aspect of my life.
Uh-huh.
I also like yoga, and so
Wow.
I do a lot of that.
Uh-huh.
I have phenomenal teachers here in Carbo, and also Tai Chi. Tai Chi is something new that I picked up, and I really like it. It's something special.
Okay, nice to know about your extra other things what you are doing. And like how big is your lab, like how many grad students, postdocs, and other like technicians and all, how does it
So now we have around 6 graduate students,
that's nice.
two research professors,
Uh-huh.
and I think 2 postdocs, one lab manager, and several technicians. So it's quite big, but it's very fluid. So we do have change in personnel quite a bit. We have
Uh-huh.
many projects going on, and because of that, there are different people at different stages of their career. They come, they enjoy the lab, and then move further in their career ladder. So
Wow, that's good. You are being very helpful. And like how do you like usually a PhD takes about 5+ years and the postdoc several years. How do you motivate these people in your lab?
oh, I think they are self-motivated. I mean, it's. I think the projects are quite exciting, and
Uh-huh.
it's all again. It's all about science.
Yeah.
If people find it exciting, they will go for it. It's like any hobby that you have. Science is no more if you treat it more than a hobby. I mean, if you treat it as a job, it
Yeah.
becomes a job. But if you treat it as a hobby, you will put all your efforts into it. And so I also have been extremely fortunate with people who have been geeks. Basically, they love science, and they just come and they help. We just work on problems, and we like that. It's everything is a challenge. And so as a result, people who come through my lab, at least the students have done really remarkably well. So many of them published at least 11 papers. Some of the amazing students published like 17, 20 papers during 5-year graduate school.
Wow.
Yeah, and these are works, some of them are works not only computational, but computational and experimental. So some of the students, they try to combine expertise and learn basically, and it's quite a unique niche for them to actually be able to do both, to bridge to these two worlds, computational and experimental. Because for many computational people, experimental world sounds a little bit abstract and detached, while for experimental people, computational sounds a little bit crazy too. So it's, but there are people, these people, they're actually learning how to marry these two fields, and it's really fun because, you know, you do something and then you go in with your hands, you test what you've done.
Yeah. No, this is a unique opportunity for the students where, like, I'm a biologist by training and all, so I don't know much about computational, but certainly the field is, if you want to learn more about the field, make very good contribution to science, like with the problems like you are looking into, like ALS, all this protein folding, biologists cannot achieve everything. So you need that collaboration, and you bring that unique set of skills to the table, which I think grad students, postdoc like it, and which is very good for science per se. So, and go ahead.
No, thank you. I said,
Yeah, and how is the interaction? Like, I mean, like, do they come up with their own problems, like based on the broad set of things, or like you give them problems? How does it go?
so we have, it's interesting. So we have main established projects in the lab that are going on, and they pose a specific, we have specific sort of directions, and within that there are quite a few interesting questions, and people usually choose what they like to do. I never press them to choose this, that, and the other. So there is a bunch of projects, and you can actually choose either of them. And usually there is something interesting, and people find something that really is fun for them. But we always have sort of a list of other projects that are going on, and you can always join other projects as well. So student learns to, or postdoc, they learn to manage their project. So you say a person picks up a project, he's the lead, but he can build, or she can build a team of people around them, because of course one person cannot have expertise in everything.
Yeah.
And our lab is quite diverse. We have physicists, we have chemists, we have medicinal chemists, biologists, so, and people with different expertise. And so what I want students and postdocs to learn is how to lead the team and build the team out of resources that you have, that you can solve the problem. And so the person picks up a problem and he or she starts building the team. So he will be the lead, but others will contribute and help. So as a result, it's beneficial for all of them.
Yep.
And so, but on the, in a reciprocal side, other people may include you for your expertise.
Yep.
And that's actually really mutually beneficial. And that's why people are so productive, and they get excited, and they learn. You know, you spend 5 years in a graduate school. It's a big chunk of your life.
Yep.
And you don't want to learn only about ALS. You want to learn about other things too, because it's exciting. And so what happens is that the students, they actually learn besides that. They have actually, they learn about other diseases or other methods or other techniques or develop tools. And it's actually exciting because when you go to a new field, new direction for postdoc, let's say, you're capable of grasping other approaches and techniques very rapidly.
And finally, like, what is the best way to contact you? Like, if somebody like looking at this interview eventually wants to come to your lab and work with you, what is the best like way to contact you?
Yeah, so the best, the best way, of course, to contact is by email. Email, I travel quite a bit, so email is probably the best way to reach me because that's gets to me every single time almost. If there are graduate students who are interested, definitely we love graduate students here. in order to join my lab, for example, or any other lab at UNC, graduate students have to be accepted by UNC as graduate students.
Yeah.
And we have a great program called BBSP, and that is an umbrella program where you join it, and you can then rotate in 3 labs and then choose one, and that's going to be your graduate lab, school lab. And so they have to be accepted by BBSP or similar programs at UNC in order to join
any lab, your lab or any other lab.
Exactly. And as a postdoc, it's just a letter. And what helps is that very clear statement of why and what's interesting to you, and CV. Yeah. I consider everybody. I mean, I definitely consider everybody.
Okay, that's good to know. If anybody is interested, they can go to the UNC website, get your email address, and write to you. And that's like, people certainly want to know what is the best form, where we can reach you as soon as possible, and all those things.
Absolutely. Yeah.
And now I think it is the final part of our whole interview is your presentation. So if you are ready, you can start the presentation. I think you can just share the presentation with the community.
All right, thank you. So I'm going to switch now. All right. And so today, then, I'll tell a story about etiology and our direction to therapeutics of Lou Gehrig's disease, or ALS. So ALS is Lou Gehrig's disease is another name for ALS in the United States. And when you start thinking about ALS, or any other disease, it's quite a complex disease. So that depending on how you look at it and where you look at it, it reminds me as the allegory from a Chinese story that it's an elephant with a bunch of wise men who are blinded. And so those blind men are looking at the elephant from various sides and trying to understand what is that creature that they're studying. And depending where they're looking, they actually see totally different things. One person sees it as a bear, another one is a snake, and the person who is touching the trunk, the foot is a tree, and so on. And that's roughly how biological view of ALS is. Depending what you study, what kind of processes you study in the disease, you basically end up in thinking that this is it. But and that's if we go to the next slide, you will see that actually, if you summarize all possible views of the disease, of what may cause the disease and the reason why it happens, all possible processes that happens in dying cells, it's a quite complex mechanism. So each of these boxes in this network presentation of this disease are various causes of various aberrant processes that happen in the dying motor neuron. And what's interesting is that, so we decided to figure out some of these processes are primary and some of them are secondary. And what we decided to figure out is maybe to get a guess what is more primary and what is secondary. And to do that, we created this graph where we point the arrow where one can cause another. And so it's a directional graph. And then we colored the nodes, the squares, based on whether how many incoming bonds that are there, or outgoing bonds are there. And turns out that there are two main big factors: is aberrant mutant superoxide dismutase interactions, is one of the genetically associated proteins with ALS that is there, and that misbehavior of that protein is one of the big ticket items here. And the other one is calcium overload that happens due to glutamate and excitotoxicity, ER stress. So there is a lot of there is calcium overload that may be, or usually is, toxic to cells. And so we decided to address Both of them are in a different way, but focusing mostly at the beginning at the aberrant interactions of misfolding of SOD. And copper zinc superoxide dismutase is an enzyme that has been identified in '93 to be genetically linked to ALS. And it's a homodimer with copper and zinc in each of the monomer. And on the left, you can see that the balls that represent residues that are mutated in the disease. So there are over 100 mutations identified that are affecting this protein. And when that happens, this protein aggregates inside motor neurons, and as a result, motor neurons die. And if motor neurons die, muscles don't exercise, they atrophy, and as a result, a person develops a disease, and unfortunately, it leads to death. So the questions that we study in the lab is why and how this protein aggregates, what is the origin of cytotoxicity, and whether we can find drugs to rescue this misfolding of SOD. And it has been an interesting journey because, and I'll tell you the good, the bad, and the ugly about this protein. So the good is that SOD is responsible for eliminating superoxide radicals, and they're elevated during oxidative stress. So you really need these proteins to avoid DNA damage and protect cells from oxidative stress. And SOD is present in high amounts in brain, which would be a problem if SOD was unstable. But on the contrary, evolution made SOD is extremely stable, much more stable than any other protein on average that we know. And so it's not clear how any genetic mutation would make it unstable. So that was not clear to us at all. And but the bad part is that SOD nevertheless aggregates, and this process occurs by first this dimer, it falls apart into monomers. Then it loses the zinc and metals and copper, and then it misfolds and aggregates. So these big things is like big aggregates inside cells that look really scary. Unfortunately, I don't have images of them, but I should show next time. Sorry. So and so for us, the puzzle was still is why that happens. Why that process of dissociation happens. And it took us a while to figure out that there are post-translational modifications on SOD, namely glutathione, so addition of this molecule glutathione that actually promotes dissociation, making this process more likely. And because of that, it is possible for SOD to dissociate. Interestingly, that molecule is activated when there is oxidative stress. So oxidative stress may potentially trigger SOD dissociation, which then triggers misfolding of SOD and formation of aggregates. But still, it's not clear what kills cells. And here's the ugly part. It took us 5 years to figure out that this SOD, upon dissociation and loss of metal, actually undergoes conformational change. And that conformational change is quite tricky and peculiar to detect. We cannot actually detect these aberrant monomers that easily, but they assemble into trimers. And these trimers are also not very stable, but we still can catch them. And it took us a while to catch them. And thanks to Yuri Lyubchenko, who actually developed this ultra-fast scanning AFM, one of the developers, he actually has this technique that allows us to catch this trimer, and you can see the images of it. And he also did volumetric measurements, and you see it's persistent for over a minute. He did volumetric measurements, and it's consistent with being a trimer. So, but then, of course, this trimer is still, it's stable for maybe a minute or few minutes, but then it still dissociates and then forms this, or associates further and forms this big oligomers. But what's interesting is that this trimer is actually, it turns out to be associated with the toxicity. And there has been an antibody developed that basically detects misfolded SOD, and that antibody, a C4F6 antibody, it was it was used to detect misfolded SOD in spinal cords of the patients. And so we used this antibody and looked at various SOD species, which is a dimer, native dimer, void volume, and then various oligomers. So oligomer 1, for example, is a trimer. Oligomer 2 is probably a tetramer. But oligomer 1 was probably the most prominent one. And this oligomer 1 lights up and recognized by this antibody, so which means that it presents something that is similar, that is what is found in the spinal cords of the patients. So it presents very similar epitopes. So this trimer presents something that is looking quite like in the spinal cords of the patient. It's called the SOD spinal cords of the patients. Now, what we also know is that because A4V is promoting the formation of the trimer, you can actually see that there are a lot of more dead cells which are stained by pink color than the blue color, than in wild type. So in wild type, there are more surviving cells than in A4V, which is the mutant associated, one of the aggressive mutants associated with ALS. And you see there is a lot of more pink, so it means dying cells due to this mutation. So the question is how. So what we decided, so the questions that we're pursuing now is how do SOD trimers kill neurons, and what are the atomic structures of the trimers. And so having said that, we got some ideas of the structure of the trimers, and that has been just published in this paper. What's interesting about it is that because we roughly know the structure of the trimer, we can stabilize the trimer. So we can modify this SOD so that it's more stable in trimeric form. So if we do that and put it back in cells, cells will die really rapidly, even more aggressively than A4V. And that actually tells us that if you stabilize the trimer, then you make it more toxic. But there are still quite a few open questions. So it's still not clear how SOD trimers kill neurons. So what happens downstream? So how does this trimeric SOD actually kill cells? And that's what we're trying to understand by doing pull-down experiments where we know how to bait for this trimer and pull down with this bait all possible interaction partners. And by knowing interaction partners, we may be able to delineate pathways that lead to cell death. And that's what's our big effort right now in the lab. Another interesting thing, we discovered another type of post-translational modification, which is phosphorylation. And phosphorylation doesn't happen for no reason in biology, and that's why it's quite interesting. And it's not clear what its primary role, why that happens, and what pathways it activates. We don't know anything about phosphorylation. And we started studying it, and we're trying to understand what happens if you phosphorylate SOD. And we couldn't find the kinase. Despite working on it for several years, we haven't identified the kinase that phosphorylates SOD. But what we did instead, we made the mutants that made this SOD, made phosphomimic, basically make SOD looks like it has a phosphorylation. And what that did, which is was surprising to us, it was stabilizing SOD. So it's almost like a nature's break. When there is oxidative stress, it gets glutathione. Maybe it's a nature's break to prevent SOD from misfolding. And we are currently investigating this event. And we are also trying to figure out what triggers phosphorylation and what are the kinases that are responsible for that. Now, in terms of therapeutic strategies, we have 3 directions. One of them, we want to fight the disease at its root. It is a complex disease, so even if we fail in fighting the disease, we want to at least find a way to prolong life. And also, ALS is a very complex disease, so we want to also learn how to diagnose it early enough, because by the time we diagnose, the motor neurons are dead, and it's too late to do much about it. And so what we want to do is to diagnose it early and then fight the disease at its roots, and then if that's failing to prolong patient's life. So how can we fight the disease? We are thinking of two approaches. One of them is to stabilize the dimer, and that's perhaps the most, at the moment, advanced part in terms of therapeutics in our lab development, where we actually screen small molecules and identified a couple of molecules that we want to actually, we have tested both in biochemical assays and in cells, and three of them show promise so far, but it's too early to talk about them. And that's strategy number one. But the strategy number two is, because we know the structure of the trimer roughly, we want to stick small molecule in between and try to destabilize this trimer. And so that's going to be strategy number two. Of course, it's better to target something earlier in the pathway, but we may need to do something at the later as well. So in order to prolong life, one way to deal with the end stage of of motor neurons is to potentially deal with calcium overload. And Shafiq Karam, who was a doctor at UNC now, he moved to Portland, Oregon. We study how can we target calcium overload with vitamin D. Turns out the vitamin D activates a bunch of proteins that scavenge calcium, and it may prove beneficial to the disease. And in this regard, we have some results that may support that in general, and they show some preliminary protection. Preliminary protection by vitamin D. So in terms of diagnostics, one interesting thing is that for the first part of the reaction to occur, for dimer to dissociate, one thing that promotes it is glutathione, addition of this moiety glutathione. And we can detect this from mass spec, and actually we can isolate this protein from blood, so we don't really need brain or spinal fluid. All we need is blood from the patients. So, and that's what we're actually currently working, is trying to figure out whether if we take blood from patients, these post-translational modifications are enriched in these patients. And if this is the case, then it's more or less a straightforward way to have a biomarker for a disease, which is right now is absent. There is no biomarkers right now for ALS. And that will help immensely diagnosis of the disease at early stages. Just a small segue is about virtual screening or drug screening. And it's a technique that we have developed in the lab that is quite, I think, fascinating to me, because it allows us to both to do docking of small molecules into proteins and figure out which ones of them will stick to them. And drug screening is, virtual drug screening has been on the market for ages. It's an old technique, but it has a very patchy history. And the reason for this is because it's very hard to model protein flexibility and the way protein interacts with small molecules by changing its shape. Not only small molecule changes its shape, but also the protein changes its shape. And so we learned how to mimic a change the shape of the small molecules, so exploring all the library of moves of the small molecules, and then perform docking, where we actually identify some site where the small molecule will be docked. We would put it there in a rigid way first, and then cluster it and see which ones would look better there, and then perform fully flexible docking, where all of these residues, the side chains, they would move and adopt to this drug, and then score these various drug conformations. And based on that, we actually can look at actual virtual screening of small molecules. This is one of the targets, and you can see how pocket where the drug that we put changes depending on the drug that we put. So it's very important to model protein flexibility. And and that's basically the technique that we are using right now to identify small molecules for SOD. So that's basically the whole story. Again, the questions that we are still targeting are this. We are trying to understand how SOD trimers kill neurons, so trying to figure out downstream pathways and ways to leverage this aggregation process through either understanding phosphorylation and upregulating proteins that do that, kinases that do that, or finding small molecules that target dimer dissociation from dissociating or trimer from associating. That's it. Thank you so much.
Thank you. Can you unshare this presentation? Okay. Thank you so much. It is a pretty good presentation.
Thank you.
I have a question. Like in the cells, like the SOD trimers are the ones which form all these fibrils and precipitation within the cells. So according to your data, so what portion of it becomes trimers in the process? Like eventually it is the trimers which is happening in the cells?
Well, the trimers actually are very transient species. They appear and disappear very rapidly. They are not present in high amounts, and that's why they present such a big difficulty for studying them and searching for them. We are not the only one who's seen them before. So Markland also seen them, and other people. So it's there are oligomers that appear, but figuring out that they're actually toxic, that's actually quite interesting, because the field, especially also in Alzheimer's field, people have thought of large oligomers or large aggregates that are toxic to cells, and they look scary actually under EM. That's why I'm sort of regretting now that I didn't put some of those images. They look scary. I mean, you would think that those big guys, they would probably create a havoc in living cells. But frankly, cells know how to deal with them. And it's possible that that this large species, large aggregates, may be actually a sink that takes away these toxic ones, and they're actually good or sort of beneficial to the organism. And in fact, there is evidence of that in yeast, at least. It has been shown that by Susan Lindquist that these aggregates actually can be beneficial for cells. So we don't know about big aggregates, but the small ones at the top, the culprits actually quite small and transient, and catching them is a thing. It's hard.
And in the cell, like what makes them to trimerize and all those things is kind of still unknown, or like you have some ideas on what happens?
Well, what triggers they would trimers form out of misfolded SOD. The question is how the misfolded SOD happens.
Yeah.
And misfolded SOD happens because SOD gets potentially post-translationally modified by glutathione, and that triggers its dissociation. And when it dissociates, it loses metals, and as soon as it loses metals, it becomes completely flexible and malleable and undergoes conformational change.
Okay. So yeah, like SOD has been there for a long time. I also did some work long time ago on SOD and all. So it's good to know this trimerization and all those stuff. So which is like, I saw your paper in PNS, and I was really like, it's a PNS paper, right? The trimerization thing recently. So I got really interested. Yeah, I got really interested, and it's very nice presentation. So it helped me learning more than like what I knew earlier. So thank you very much. I really appreciate your time and effort. And maybe we will catch up sometime in few more months. Whenever you have something new, you can share with everybody through, again, Science Hangout. So again, I appreciate your taking time out and coming and talking to us.
Thank you.
Thank you so much.
Thank you. Bye.
Bye.